Hello everyone, it is time for Lecture 2, and Lecture 2 covers the topics you see on your screen. So I want to get into it. Be aware there will be some math later, and that might take some of your attention. You might need to work through some of it, depending on how recently you've seen these things in a math class. That'll be okay. You'll be able to do it. We just have to get through it together. So we will get there. First, I want to talk about cell structures and morphology and growth. So we're going to start with morphology, and that is the shape of bacterial cells, what they look like under the microscope. And in these slides, I'm going to give you some examples of bacteria that have these shapes, but I'm not going to ask you to memorize that. It's just that if you already have heard of these bacteria, now you will see their shapes. We will talk about a lot of specific bacteria and the things they do, especially in Unit 4 when we look at infectious diseases and the pathophysiology of infectious diseases. But for now, we're more interested in general traits that the microorganisms have. One very common group are the cocci. These are spherical bacteria. They typically don't move much, or they don't swim through water. They just grow where they are, and that's something that works very well for a lot of bacteria. So we would call one of these cells a coccus, and more than one would be a cocci. These come in different variations, and those variations have to do with how they stay stuck together after the cells divide. And different species will do this differently. So let's just put all these here. So the biggest, the most obvious one to me is the staphylococci. These are like clusters of grapes. They are randomly associated. So when they divide, they're kind of sticky. They stick to each other, but they don't have any organized three-dimensional pattern. Some that do have an organized three-dimensional pattern are the streptococci. These keep track of the plane that they separate by. So when two daughter cells separate, they're going to remember the direction they went in, and they will just keep making this chain longer. And it's an interesting process because each one of these cells will divide. So this one will divide, but this one will also divide, as will this one. So the chain gets longer from the ends and from the inside. So streptococci do keep track of the plane of division. Sarcinae, so these little packets, they stay in groups of 8. And what's happening here is they divide once along one plane, and then the daughter cells divide in a perpendicular plane to that. And then their daughter cells divide in a perpendicular plane to that. And after 3 divisions, they end up with a cluster of 8. That I think you can see there are 3 different sort of planes you can separate these on. So these are things we will see. I think most common will be things like diplococci, where 2 of them stay stuck together, streptococci and staphylococci. One thing that might throw you a little bit as a new microbiology student is that staphylococcus or staphylococci is both a shape, but it is also a genus. And that's because this genus was discovered and it had a notable morphology. And that was back before any of us was born. Long, long time ago. So that is a little bit confusing because there can be staphylococci bacteria that are not in the genus Staphylococcus. And we see the same thing with streptococcus. There are streptococci growing bacteria, they grow in that shape, but they're not from the genus Streptococcus. So what these all have in common is a roughly spherical cell shape that we call a coccus. And the plural again is cocci. Equally common, maybe more common, is the shape bacillus or rod, so a little staff or stick. And these are cells that will elongate notably before they divide. So they'll grow longer and longer before they divide. The plural is bacilli, and they can also appear in groups. Like some of them will stick together with the other daughter cell and become diplobacilli. Others will divide in chains. But we think about the individual cells. I've never heard anybody say streptobacilli or strepto or diplobacilli except in a textbook. Those words have more to do with the cocci, realistically. But what I do want to point out is that some of them can grow as filaments. So they grow in really, really long chains that would have like 100 cells in a row. More important than that is that the length of these differs. They all have roughly the same thickness for whatever reason. They're all roughly between half a micrometer and 1 micrometer thick, roughly. But they could vary in their ratio of length to width. And that is sometimes that helps us identify them, as I'll get to later. So these are very common, and in cases where they grow as single cells, a lot of these will be motile. That is, they can swim through water. Others might be motile in different ways, where they can sort of crawl along surfaces. Let's see. The last major group would be spirals, and these are kind of also common in nature. So there are 3 sort of different defined spiral-shaped bacteria based on whether they have one incomplete turn or twist, a small number of twists, or a lot of twists. And these are some very specific things that we find in nature. The vibrios will be kind of short and curved rods, and Vibrio cholerae is a very famous one. It's named after its cell morphology, and it causes cholera. Spirilla, or one single spirillum, is a cell with like 2 twists like this, and there are several different species that grow this way. Each of them is called a spirillum. And then spirochetes are very specific. There's like one group of spirochetes that has a few genera in it. It's a phylum, actually. There is one phylum of spirochetes. And these 2 are in it. Borrelia burgdorferi causes Lyme disease, and Treponema pallidum causes syphilis. And they're both spirochetes. They're long and very skinny. And the way this is drawn here, it looks skinnier than these 2, and that is a real thing. These are drawn pretty much to scale. The only thing I would change if I was making sure these were to scale is the spirochete would be even longer. So they are long and they're unusually skinny. Okay. So those are the possibilities in cell morphology, that is the shape of individual cells we can see under the microscope. And we don't these days learn anything about bacteria by looking at their shape under the microscope. But in the early days, this was the best we could do. I also want to talk a little bit about the common confusion that happens because of the historical use of microscopes and cell morphology to describe bacteria. So these days, we think a lot more about the genetics of the bacteria, what their DNA is, what proteins they can make, what their behaviors. We think about that all the time. We don't think as often about their shapes. But originally, again, the first thing people could see about a bacterium was its shape, was its morphology and its arrangement. And so some of the most common or commonly found on humans bacteria were named after their shapes, and those include the genus Streptococcus. And we will learn in Chapter 4, or Unit 4, sorry, we'll learn about Streptococcus pyogenes, or group A strep, and Streptococcus pneumoniae that causes both of them cause a whole lot of diseases. And Staphylococcus, genus Staphylococcus, same thing. Staphylococcus aureus is really spectacular in that it seems to be able to infect any tissue in a human body. And it commonly lives in people's noses. And Staphylococcus epidermidis is common on people's skin and some other things we'll talk about. And then there's a whole genus of Vibrio, and the bacterium that causes cholera, Vibrio cholerae, is named after that. And these are all just named after their shape and arrangement. So now would be a great time for you to pause the video and draw a Streptococcus and a Staphylococcus and a Vibrio. And what you'll see, I think, maybe later in these videos is how to write a genus name and how to write a species name. And so you'll notice when I wrote these genera, that is the plural form of genus, I put them in italics and capitalized the genus. And then later when I wrote the species name, I abbreviate the genus and I write the species name not capitalized. So if you read the word Streptococcus and you're wondering, is this talking about a cell morphology and arrangement, or is it talking about a genus of bacteria? The formatting will tell you, because if it's capitalized and italicized, it's a genus, and if it is not either of those things, it is a cell morphology and arrangement. I do want to point out the variations we see in cell morphology or shape, and these are kind of beyond what we understand right now. But within a single species, they could have slightly different morphology depending on whether they're growing in liquid broth or on the surface of an agar petri dish, or if you find them outside in soil or water. And so these are some of the variations or non-variations. So the length of rods can vary in these environments, but the thickness does not vary. So if you take one specific species that can grow as a rod, it may have different lengths in different environments, but its thickness will be the same in all those environments. So rod-shaped bacteria or bacilli, this one, if you had grown it in a different environment, it might be shorter, like much shorter in some cases. And then, sorry about that. And sometimes I've seen growth in chains be really variable, again, depending on whether they are, depending on how they're growing. So there are some, well, even Streptococcus bacteria, they won't grow in chains in liquid broth in some situations. So those groupings we see are not always diagnostic. But the cell shapes, like, is it a bacillus? Is it a coccus? Those tend to be very stable and you can be sure of what you're looking at. And the curvature of these curved ones is not, from what I know, is not variable in any meaningful way. So you'll always recognize a spirillum or a spirochete. Spirochetes are so thin that you sometimes can't see them under the microscope with a light microscope. They're almost invisible. Okay, that was cell morphology, and I'm going to end this first video there so that we can really dive in headfirst to bacterial growth, which is going to be a recurring theme throughout the rest of the term. So I will see you there.